Now, in trying to make a diagnosis in various fields or for various reasons, you might actually want to pinpoint a microorganism that is causing the issue. Do you understand? So one of the ways of doing this is, for the little DNA you have, you want to amplify it. It's like you want to increase its loudness so that it can better pinpoint the microorganism causing the issue. So you can do this through several techniques, okay, being used in diagnosis and all. So for the different techniques for amplifying DNA, let's introduce it. So they're actually important tools in molecular biology. So they enable researchers to be able to generate large quantities of DNA just from the small ones that they have. Okay. So the first one is this, which is polymerase chain reaction. It's the most widely used method. It was introduced in the 1980s. So the cycles of denaturation, annealing, and extension using DNA polymerase. You can pause this to look at the picture, right? So variants, we have the conventional type, we have the quantitative type, we have the reverse transcription PCR type. Okay. So moving forward, then we have the isothermal amplification. Then we have the multiple displacement amplification. So let's look at the isothermal amplification now. It amplifies DNA at a constant temperature, okay, eliminating the need for thermal circulation. So types, we have the loop mediator type, we have the helicase dependent type. Okay, can pause to observe the picture. Then we have the multiple displacement amplification. It uses a special DNA polymerase with strand displacement activity. It's ideal for whole genome amplification from small samples. I've seen it. Then we have the rolling cycle amplification. So amplify circular DNA templates. It's used in diagnostics and also nanotology. Rolling cycle amplification. Now for up next, we have the bridge amplification. Okay, we can also pause to look at the picture. So for the bridge amplification, it's used in Illumina sequencing platforms. Okay, so DNA fragments hybridize to oligonucleotides on a flow cell and form bridges for replication. Then we have the emulsification PCR. Okay, then emulsification PCR. All right. So why do you actually need to, amplify this DNA? So gene expression analysis, genetic diagnosis. So you want to make diagnosis from the cellular level. Forensic DNA profiling, environmental and microbial studies, then next generation sequencing preparation. Okay, so that's what we have here for these different techniques that you use to amplify DNA.